Hi, thank you for having me. This is work done with a colleague in our lab, Yuting, who is unable to be here today, but she's got the poster on the virtual platform and a short talk on the virtual platform as well. So please go check that out. So in this talk, we're interested in phylogenetic comparative methods. So in this case, given a species tree and the counts of the protein coding repertoire in each leaf species, what we're interested in doing is trying to find the ancestral protein coding repertoire and the families that are gained and lost on each branch of the species tree. So how we do this depends on the method used, the different assumptions about the evolutionary processes, and the constraints that are applied. So one common approach is to use a parsimony model. In this case, we assume changes are rare, and we're looking to minimize the number of gain and loss events. A popular parsimony approach is the Dalo parsimony, which imposes a further restriction that a character can only be gained once but lost multiple times. And this approach has been used quite often in the literature, and many previous studies with Dalo have reported widespread gene loss and streamlining in genomes. Another approach is a probabilistic approach, in which case we're going to model evolutionary change as a stochastic process with rate parameters. And in the case of maximum likelihood, we're going to maximize the likelihood of observing the present day repertoire. And in particular, we are going to look in this study at a birth-death gain model. So I'll refer to this as a BDG model. In this case, the evolution of character states are modeled using a Markov model on each branch of the species tree with gain, duplication, and loss events. And in this case, we're going to be inferring rates as well as events and ancestral states. So in this work, we looked at investigating the evolution of the protein repertoire in 21 holozoan species shown here. So this is Metazoa and the two most closely related single cell species in choanoflagellates. And in particular, I'm interested in domains and domain family evolution. And so we're going to represent the protein repertoire using the superfamily database of domains. For those that are not aware, domain is a protein module, and they're thought to have distinct structure and function. So I'm showing here, let's see, the pointer. Showing here is the Sark kinase protein in humans, and you see the different domains are color-coded. So we have an SH3, SH2, and a kinase domain, and the schematic of what this protein looks like here. And just to note, these are thought to have distinct functions. So the kinase is enzymatic function. It's going to be phosphorylating a tyrosine residue, and SH3 and SH2 are binding proteins. So they bind different parts of proteins. So they act for protein-protein interaction, as well as for self-bind to other areas of the Sark kinase. So they act for activation and activation of the protein as a whole. And so the model we used was the BDG model implemented in count. And what we can get from this is the expected gain, loss, expansion, and contraction of each domain family on each branch of the species tree, the expected size of the family in ancestral species, as well as the domain gain, loss, and duplication rates. So I'm going to go through some aspects of the results from this study. So for here, I'm just going to walk you through. We can get expected family gains and losses. So in red outline, that's the number of domain families that were lost. In a green outline is the domain families that were gained. So they weren't present and are now present. And then I'll show in solid the net change. And we can also look at family expansion and contraction. So families getting bigger and smaller. Again, similarly, green outline is expansion. Pink outline is contraction, and then solid is the net change. And what I want to point out is that what we can see, is we can see a number of different approaches on how domain families evolve. So we can see remodeling and specialization, as well as expansion and streamlining. And what I want to point out is if we group these together, what we see is a continual remodeling of the protein repertoire that suggests that we have substantial genomic plasticity. So what we see is specialization, where we're losing some domain families and the domain families that are retained are expanding, or remodeling, where we're seeing a little bit of gain and loss of domain families, but it's pretty equal extent. And then again, the main families that are being retained are expanding. And this is in contrast with the role of genome streamlining seems to be much more modest, at least in proteins. And we only see this in a few genomes, and these are known to be smaller genomes as well. So this is a kind of in stark contrast to what's been reported extensively in the literature, where we see lots of widespread contraction. Okay, 3 minutes left. So the next part of this is a model comparison, which we don't really have time for. So come see us at the poster, or come see our poster on the virtual platform. But one of the other things I really wanted to talk about with function today is looking at rates of domain duplication and loss, and how are they related to function. And so what we did is, for our domains, we had to find functional annotation, and this is a little hard. But in 2004, Vogel and colleagues worked to annotate all of the domains in the superfamily database. They looked at 50 detailed functional categories they defined, and then annotated each domain with one of these functional categories. And they did this by looking through the literature and manually curating this. If we look then, just a short, quick story here. If we look at sorting by duplication rate, and we see the different functional categories on our x-axis. So we see things that have evolved slowly are things that you would expect. So they're associated with basic cellular processes such as transcription and translation, while things that are evolving with faster rates tend to be things with Metazoan innovation and things you might expect to have evolved more quickly, such as immune response. But I wanted to bring a note to everybody in the functional group. So we use this Vogel annotation. There are other ways of doing domain annotation. Of one particular note, InterPro2Go is working on annotating all the domains at InterPro. However, at this point, so if you look at the superfamily database, only about 36% of domains have been annotated. And for PFAM, only about 25% of PFAM entries have been annotated. There is other work that has been done to look at automatic transfer of Go annotations from proteins to domains. And so there hasn't really been a big discussion of whether this is an appropriate way to annotate domain functions. So for example, right, you're looking across all the species, all proteins that a domain is found in, and then finding terms that are enriched. And then assigning the domain that function. And is that an appropriate way to do this? Is it not all proteins have that function annotated with it? So should the domain get like a partial score for each annotation? There's a lot of discussion on what would be the most appropriate thing here. And on that, I'd like to conclude and tell you to please come see our poster and talk more about function annotation with domains. And we also have a paper on bioRxiv. Am I mic'd? There you go. Why don't you set up and we'll have time for one or two questions for Maureen in the meantime.
Should be going over.
You mentioned several types of events, including contraction, whereas your Markov chain has just birth and death.
And it's birth, death, and gain. So you can gain a new family, and then birth is loss of a single copy. So sometimes that loss is a contraction of the family, and if it's the last copy loss, it's
Contraction is under losses.
Yes,
Okay, understood.
yes.
Thank you.
Thank you for the very nice talk. Do you see any impact of genome quality, genome annotation quality? Because I saw the landscape, the Amphioxus in there, and we worked on this, and the previous genome and the new genome had a huge difference in the rate we could measure gene duplication and loss. So
This was Amphioxus. Which was your?
landscape is Amphioxus.
Yes.
I'm not specifically asking about Amphioxus, you don't need to know all the species. But is there an impact of genome quality?
Yes. So there's genome quality, there's genome annotation, as well as domain annotation of the genomes. For the Amphioxus, that is the one we did some in-house, and you can read about it in the paper, but we did some in-house fixing of that annotation. We did the analysis here. But yeah, it can make a very big difference. I think for anything when we're doing a comparative method such as this, the annotations count. We did try to find genomes that were supposedly well annotated and well sequenced.
Let's thank the speaker.